Cancer Institute USA, and also visiting faculty at Johns Hopkins. His interest is in the development of advanced technology in imaging using 3D electron microscopy, a very hot topic, and he applies this to HIV, AIDS, and cancer research, and we're looking forward to his talk. Sriram, go ahead.
Thank you, Sabina. It's very good to see you after all these years. Close to 2 angstrom resolution, 15 superposed on the melting polar ice cap. And the point was that these methods were being increasingly used, and our work had shown that you can actually get essentially atomic resolution with cryo-EM. One of the changes in cryo-EM the last decade is the ability to investigate structures of smaller things. Cryo-EM as a discipline itself is not new. Been used by a number of scientists at the lab for well over a decade. But while 10 years ago, we could investigate the structure of the large entities, like icosahedral viruses, the large protein complexes, ribosomes or proteasomes, in more recent work, the fact that we can now extend this to look at much smaller and especially dynamic protein complexes, it makes it very exciting or very broad range of problems of interest in cell biology. I will mainly speak about some of these, the work on these smaller complexes, which is where most of my own focus has been. And I'd like to begin with a brief video that introduces the method to those of you that may not be familiar with how it works. On the face of it, it looks very easy. Essentially, it begins with a purified protein, or purified virus, or whole cells in many applications. This is delivered to the surface of an electron microscope grid that's about 3 millimeters across, plotted with Whatman #1 filter paper that most organic chemistry labs have. And at this point, the protein or the suspension is in an aqueous buffer, which is sponge frozen in liquid ethane that's cooled by liquid nitrogen. At this point, we vitrify the sample, we freeze it in its tracks, and it can then be delivered into the column of an electron microscope via a cassette, which is also cooled to liquid nitrogen temperature. Once it goes into the column of an electron microscope, we can irradiate this with an electron beam, collect images that are essentially projection images of these interventions. So each of these structures you see here are images of the protein, and one can then computationally process these images to reconstruct the 3D structure. So many of these data sets are somewhat automated, and I think the excitement is that methods can now potentially be very generally used. Although we've been working on cryo-EM for some time with the development of new detectors, we essentially showed 2015, that's 2 years ago, that one could begin to use cryo-EM methods go past what many of us thought would be a sound barrier cryo-EM technology, which is about 3.5 angstrom. We showed that we could get to these really high resolutions where you can actually visualize densities with side chains directly. So at these kinds of resolutions, for those of you who are used to looking at crystallographic density map, this is essentially an EM density map, where you can visualize densities with side chains directly. You can place them into the density map. And I think the fact that we can now visualize unambiguously these side chains is very exciting. In addition to visualizing the side chains, for example, with aromatic rings, we could clearly visualize fold in aromatic residues, which typically are associated with structures at resolution of angstroms or thereabouts. But perhaps the most exciting finding was that we could visualize density water molecules. The idea that we could visualize water molecules in a cryo-EM structure was actually very surprising to us and, of course, transformative, because at these kinds at resolution that you can see water molecules, it becomes possible to now look at chemistry of the structure itself, as well as chemistry of small molecules bound to the protein. And this was my main interest, which was to begin to localize the binding sites of small molecules that actually bind to the protein. In this case, G is a molecule that inhibits beta-galactosidase. And not only could we show that it was bound, we could also show density for a single water molecule between PETG and the active site glutamic acid residue. Last year, we extended this to real-life applications, and this was always the goal. And in work we did with a cancer target, a AAA ATPase called P97, we demonstrated that we could use cryo-EM methods to achieve high-resolution structure for P97, not only by itself, but in complex with a molecule being developed at the National Cancer Institute as a possible inhibitor of this enzyme. So this was the very last year when this paper was published. This was the first example of the application of cryo-EM to a clinical target, and also the first example where we showed that a small molecule could be visualized at essentially atomic resolution when bound to a protein target such as P97. The reason this was interesting technically is that P97 has been known for a long time. It's a very important protein. It's involved in protein degradation, it's involved in autophagy, and many other important roles. And over a decade ago, work at Stanford has shown that the structure of the protein in the ADP bound state, adenosine diphosphate, the structure of the protein, it's a hexamer, and it has these three domains that you see colored on the right. The central question in the field was the nature of the conformation changes that take place when ADP is exchanged for ATP. And this remained structurally a puzzle because it was almost impossible to crystallize the protein in the ATP. And although there were many structures reported with analogs, it was clear that the changes were not being captured in the crystallographic studies. And we showed last year in this paper that I briefly mentioned before that we now understood the origin of this problem. And this is demonstrated in this model, which shows structure in the ADP bound state. And what we showed is that when the first molecule of ADP is exchanged with ATP gamma S, there's a change in one of the nucleotide binding domains that's shown in magenta. But when the second molecule is exchanged, there's a much larger structural change, leading to an opening out of the N-terminal. Now, the reason why it's difficult to see all of this with crystallography is these three conformations are present simultaneously in solution. And we showed that it is possible to computationally separate three conformations and solve each of them at atomic resolution. So now we have these three structures, each done at atomic resolution. But our interest was, in addition to figuring out the mechanism, to be able to map the molecules at high resolution. And the molecule you see up on the upper left, the phenylindole derivative, that at that time had been shown to have specific activity against P97 ATPase, at least the P97 itself, versus other ATPases. But we got very lucky in the sense that we demonstrated the binding of this molecule freezes the protein in its tracks, and allowed us to determine, as I said before, the very first high-resolution structure by cryo-EM of a small molecule bound to a protein target, and begin to get information on the detailed interactions in the binding site. This is the kind of thing that crystallographers have been doing for 2 decades or more, but it's exciting that now we can get to this level of information with cryo-EM technology. We continued this work last year, trying to further advance the boundaries of resolution that could be achieved for and also to begin to push the boundary of these sizes. We always wondered, would cryo-EM be acceptable to look at smaller proteins? And in this paper there, where we reported advances on both fronts, we took on a set of proteins which are dehydrogenases, glutamate dehydrogenase, isocitrate dehydrogenase, and lactate dehydrogenase, that's in the middle. And the transition from glutamate to lactate to isocitrate dehydrogenase essentially is is a progression in size that gets smaller and smaller. And IDH, isocitrate dehydrogenase one in particular, is now below 100 kilodaltons. And this was a huge milestone for us to get to. Because if you look at this metabolic pathways chart that almost all of you are familiar with, actually used to be in the corridors of Govind's lab at MIT, is where I saw it for the first time. The goal really is to have these metabolic pathways come alive in 3D, because although we know the identities of many of the molecules in this chart, we also like to know what this looks like in 3D. That drove much of the work in my lab for decades. But now in the context of cancer and other diseases, where many of these proteins are affected or mutated, what we seek to understand now is the way in which these molecules might inhibit specific enzymes in various aspects, involved in various aspects of metabolism. This work, which we reported last year in Cell, also enabled us to push the advanced resolution achieved by cryo-EM, where we showed that glutamate dehydrogenase could now be resolved at 1.8 angstrom resolution, and this still today is the highest resolution structure reported by cryo-EM in the electron microscopy data bank. At this resolution, you can see densities of side chains, as I showed before, as well as densities on water molecules, many of them that you see circled here. The more important thing we were after from a clinical perspective was to demonstrate that we could visualize a small molecule being developed at the NIH called ML309 that specifically targets a mutant form of isocitrate dehydrogenase. And the reason this is important is that patients that have mutations at this particular residue have cancer that is due to the change in metabolism. So instead of isocitrate producing alpha ketoglutamate, which is what it does in normal cells, in the case of patients that carry this mutation, it in turn produces 2-hydroxyglutamate, which is an oncometabolite. So the task of finding molecules that inhibit IDH1 is to find ones that specifically inhibit the mutant form, but not the wild type form. And there has been success on these things, including success at the NIH, as well as 2 other commercial entities that have reported success in molecules that block IDH1. Now, for the first time, we show that we can use cryo-EM methods to go after locating a small molecule in IDH1. And it's significant because this has not yielded to crystallographic analysis for reasons, the same reasons as with 97. Once the molecule binds, it's in equilibrium between 2 states and it's both present at the same time. We can now computate these up. So this also demonstrated that cryo-EM could now be used to study proteins smaller than 100 kilodaltons, which many of us thought even last year would be almost impossible. The application to membrane proteins, which I learned about really for the first time in MIT, has also been very interesting, and worldwide there is tremendous interest in applying these methods to study membrane proteins, in particular because they can be challenging to study by crystallography. Among the proteins we chose to work on, we focused on ion channel. And one example of this is work on ionotropic glutamate receptors, work that we did with Mark Mayer at the NIH. And as many of you will know, these are critical proteins in the brain. When they don't work properly, it can lead to diseases such as Alzheimer's, memory loss, Parkinson's, and many others. And there is therefore great commercial interest in finding molecules that might target one or the other subtype of these ion channels. Eric Guo, who also was in Govind's lab about the time that I was there, reported a seminal piece of work in 2008 or 2009, where he showed the structure of the resting state, the AMPA receptor. And from that point onwards, the challenge in the field for the next decade was to decipher the nature of the gating cycle. What actually happens when glutamate binds, and how is this relevant to the transport of ions across the membrane? And in a series of papers that we published along with Mark Mayer, first in 2014, and then second in 2016, we demonstrated that using cryo-EM methods, we could determine the nature of structural changes that resulted from glutamate binding to activate the receptor. But even more interestingly, we also demonstrate the changes that took place with desensitization of the receptor. These two processes of activation and desensitization are processes that happen in milliseconds. And understanding their structural features is, of course, critical to drug discovery. And the work we reported last year is also technically significant because not only is it the first, and to my knowledge, the only atomic resolution model for the desensitized state of a glutamate receptor, we also showed that we could visualize densities for carbohydrates that you see on the lower right. Each of those are N-linked sugars that we can visualize density for directly close to the protein. And unlike crystallographic analyses, we don't need to truncate these residues, or to truncate the sugars or mutate the residues, because we're just averaging the structures of the molecules. And if it's disordered, it doesn't get averaged. And whatever is ordered and close to the asparagine residue shows up in the density map. Among the other things that showed up in these studies was a very interesting and surprising finding that I'd like to share with you here, which is the discovery that desensitization leads to a very large structural change. We all know examples of allosteric proteins with very large changes. But what was initially puzzling about this was the fact that typical cross-linking studies had shown that the same residues were cross-linked in resting state and desensitized state. And yet, we were finding a very large structural change. And it turns out the answer to this puzzle is that the changes are so large that the same residues are cross-linked, but in the neighboring subunits. So this is a tetramer. And it turns out the changes are so large, and the rotation is to an extent that it actually leads to a contact of the same residue one subunit over. And of course, that's why the cross-linking studies suggested the same residues were cross-linked, but it did not mean that there was no structural change. In similar studies, we worked recently on an ion channel that transports magnesium. This is a prokaryotic ion channel. It's called CoRE. And this is work we did with Eduardo Perozo at the University of Chicago. And the reason we were interested in this ion channel is because crystallographic studies had shown for several years that the structure of the protein was independent of the presence of magnesium. So the structures that you see superposed are with and without magnesium. And the surprise here to biochemists and electrophysiologists is that the changes that people observed with spin labeling studies and with other readouts implied that the magnesium binding or loss of magnesium seemed to change the protein structure a lot, but this didn't show up in the crystallographic analyses. So our guess was that the crystal structure was essentially inhibiting the true structural change. And to do this, we essentially initiated intensive collaboration with Eduardo Perozo to test the structure with cryo-EM in the presence and absence of magnesium. And our first results were to demonstrate the structure with magnesium, and indeed it showed that the structure was the same. as the crystal structures, we could visualize density with side chains, magnesium ions. It was also a technical advance for us at that time last year, because we showed that now proteins in the size range of 200 kilodaltons, membrane proteins, could be tackled with cryo-EM. The exciting discovery came when we took magnesium away. What we found, to our great surprise, was that in the absence of magnesium, the protein undergoes a very large structural change. We're talking now changes of the order of 30 angstroms that are associated with loss of symmetry. So not only does the protein change a lot in structure, there is no longer five-fold symmetry. Ever since Rod McKinnon's work, many of we were conditioned to think of symmetric opening and closing events in ion channels, or perhaps small changes. But now that we've removed the constraints of a crystal lattice, I think it could be a paradigm shift in terms of extent of change that ion channels display when they're in the context of a membrane, but without the context of a 3D crystal. The last example I'd like to share with you is work just published on the application of cryo-EM to nucleic acid protein complexes. And this work we did in close collaboration with Dinshaw Patel, who's at the Memorial Sloan Kettering Center at New York. And on the left, the schematic on the left-hand side essentially captures the structural biological problem we're trying to solve and the biology that's relevant to it. When bacteria get infected by bacteriophages, CRISPR complexes, which are now very much in the news, swing into action, and one of two outcomes is possible. Either the DNA is recognized by a CRISPR complex, and it leads to cleavage of the DNA, and the bacterium survives, or the bacterium is destroyed, and it leads to essentially it's a replication of the phage. What we showed is in a structure that actually was perhaps the most challenging we've done, the near atomic resolution model for the CRISPR-Cas complex, 9 polypeptides, no symmetry, with RNA and DNA. And what emerged from this work again is the revelation of the extraordinarily dynamic nature of this complex. And what we've shown now is that the binding of DNA leads to this dramatic change in fit. Essentially, you can think of these complexes as being in a quiescent state until DNA arrives. And when the right DNA arrives, it's recognized by the PAM motif, it opens out the structure, and then it leads to a nuclease that cleaves the DNA. We now have structures with numerous inhibitors that compete with this degradation, the phage fighting back. And now we have an understanding of the location at which these inhibitors bind. This is one such example, an inhibitor called ACR10 that was reported by Alan Davidson at Toronto. And we show that the mechanism by which this inhibitor inhibits phage, the function, is to sit at exactly the same side. The DNA binds, you see this superposition on the left, and also it leads to interesting structural changes and to essentially a clasping of the inhibitor by movement of the hook domain that you see on the right. Essentially now this opens the door for us to look at not just the effective DNA binding, but also the different ways in which inhibitors block DNA binding and thereby enable the phage to replicate. So these are three examples I've discussed with you. Work on the one hand, the metabolic protein complexes, ion channels, and nucleic acid protein complexes. And the foundation for essentially all of these example studies was laid in my time at MIT, where I was exposed to the very rich biological landscape of what's possible. And I'm, as I said before, really grateful that I had this opportunity. The cryo-EM field itself has now evolved from a time 15 years ago actually 20 years ago when I did the very first structure of beta-galactosidase with Richard Henderson, and when we had 30 angstroms resolution at the left, but we are now at essentially atomic resolution on the right side. And I want to end by pointing out that as the field grows, more and more structures are being done at high resolution. This plot shows you where the field is today. The x-axis is resolution achieved. The y-axis is size of protein. And you can see that the area which is really most interesting from a therapeutic point of view for drug discovery, we believe is to be able to look at small proteins at high resolution. This is the lower left corner. There's about a dozen entries now at high resolution by cryo-EM. I've spoken to you about many of these entries that come to work in my lab, and I think this will hopefully begin to get larger in the years to come. The last slide, I'd like to point out that cryo-EM is a broad discipline, and my own interest in this technology has been to bridge the gap, not just to look at proteins, but to look at viruses. Nearly half the work we've done in the last decade has been on HIV, leading to the first structure of the primary envelope glycoprotein, template of vaccine design. We've reported over 100 structures of HIV, influenza, and Ebola glycoproteins using a closely related method called electron tomography. We've looked at structures of whole cells, bacteria in the context of signal transduction, chemotaxis in particular. And finally, I haven't mentioned work we're doing to use these methods to look at very large structures, tissue in 3D, using a technology called focused ion beam imaging, which I originally seen in the semiconductor world, but now applied. Brought over to biology to look at tissue, particularly now main focus is to look at this in the context of cancer, to differentiate normal cells from transformed cells in the context of aging, understand changes in mitochondrial structure as we go through the aging process. My website is shown on the left-hand side, email address on the right at the bottom. And again, I'd like to thank you very much for your attention, and I'm happy to answer the question.
Please. My.
Dr. Subramaniam, I have a few queries. You showed resolutions from 3.8 to 1.8, which is amazing. My queries are, first, what was the number of particles you picked up for the class averages in each case with increasing resolution? The second was, what was the sigma contours of your electron density maps? And as far as the ligand is concerned, what was the occupancy of the ligand, and you contoured the ligand density at what sigma?
Did you get that?
Let me say it back. Right. So the first question was how many particles that went into the different class averages. Is that right? They vary. I think what we find is quality matters more than quantity. And when we reported varigalactic space at close to 2 angstroms, we had about 40,000 particles in the mix. We work with the glutamate receptor similar to 60,000 or thereabouts. So the number itself doesn't seem to be such a critical factor, but the quality of the image and the information in the Fourier domain, that's probably the most important. Your second question was something to do with the density map. Yeah, okay. Let me address that. So you're asking me what's the sigma value? So your question is, what is the sigma value at which we are showing these maps? Is that right? So let me clarify. So the maps that we get from cryo-EM are what we call electron potential maps. They're not really electron density maps, and not subject to the same sigma scaling that you might look at a 2F minus FC. What actually what we are showing are thresholds that you can loosely convert into something that is similar to what is seen in electron density map, but a direct correspondence to sigma value is actually not so meaningful. And the other factor that's quite different is these are maps that come from the data, the experimental map. There's no model used in producing them. So unlike a 2FO minus FC map, where you have phases come from a model, here the phases come from the data. And because it's protein in solution, you have variable order. And so it's closer to an NMR situation where you have proteins that are essentially dynamic. And the B factors, if you think of B factors the way you do in crystallography, the B factors vary a lot from the inside to the outside. That doesn't mean the structure is poorly resolved. So these density maps that I'm showing have B factors, and you can actually download these from the data bank, and you can apply your own B factors to look at these structures. But maybe this is the better question offline. You can send me an email.
Voltage gated ion cell in the neuron cell, ion channels in the neuron cell. So the transport of ions through the channel is under the influence of electromagnetic field, which is coming from the electrical activity from the neuron itself. So, so how such environment affects the predictions?
So your question is whether the structures we observe are influenced or will be influenced by electrochemical potential or transmembrane potential. So these are studies, the membrane protein studies I showed are with membrane proteins solubilized in lipid detergent micelles, and in some cases with amphibol, but they do not have a closed membrane, so we have no gradients. The work with HIV, which I did speak about, those were all done with intact viruses. So there, that work I foresee will be extended, looking at structures of proteins in vesicles, and there perhaps we can do the kind of experiment that you're suggesting. See you. Yes, bye.
So
Bye.
it's a great pleasure to introduce next, as the next speaker, another contemporary of mine, who is Mark Brayman. Mark was born and raised in Rochester, New York. He received his BA in 1977 from Harvard, his PhD in biophysical chemistry from the University of Berkeley, and then joined Gobin's lab Gobin's lab, actually in collaboration with Kenneth Rothschild of Boston University between '83 and '88, supported by Helen Hay Whitney Foundation Fellowship and the Lucille Markey Scholar Award. From '88 to '98, he was assistant and then associate professor at the University of Virginia Medical School, and received the Whitaker Foundation Young Investigator Award. And since 1998, Mark has been professor of chemistry at Syracuse University. Mark.
Okay, thank you, Sabina, for the introduction. Thanks to the organizers, Rajinder and Tom, for inviting me to come and speak. And also thank you to the local people, Dr. Jay and his team of wonderful students who have made the logistics so much more comfortable for a first-time visitor to India than they could have been. It's been very helpful to have their assistance at every step of the way. Let me start with one of my fondest memories of Gobind. You saw the video, he mentioned his cottage in New Hampshire and some of the trips that he invited the lab to go on up there were wonderful. I remember one time being one of the first to arrive and standing with Gobind on the deck looking out as down the hill as some more people were arriving, and out of her car came Dr. Breitenberger, Caroline Breitenberger, and one of the things she did after unloading her plastic cooler chest was putting it on her head and walking up the hill towards the house. And Gobind looked down at that and said, "Oh, Caroline's carrying that the way I used to carry water on my head in my village." And that really cemented, crystallized for me the idea of Gobind as an exemplar of not only geographic mobility, but also economic and social mobility and technological mobility in the 20th century. And I try to pay homage to that in my interactions with students who come from backgrounds that are not as privileged as mine. And that's one of the reasons that I came here, was to help make sure that I get a chance to pay some homage to that. Well, one of the recurrent themes in the structure and function of the membrane proteins that Gobind focused on is the behavior of conserved arginine residues that during the activation of these proteins undergo quite large conformational swings, including into regions of the membrane where they're pretty well excluded from most of the water that they would normally be found surrounded by. This is true not only for those membrane proteins, but also for some other membrane proteins that have been looked at lately. So, for example, voltage-gated ion channels. And even this example of one of the smallest proteins, actually just a dipeptide, that involves arginine is the endogenous brain analgesic Kyotorphin. Where do I, oops, I'm going here. This is just tyrosine arginine, and it was first isolated in 1979 from rat brains. The demonstration that it functions as an analgesic originally required that it be injected into rat brains directly because it couldn't get past the blood-brain barrier. More recently, people have modified the C-terminus of this with more lipophilic groups and have just recently shown that they can get it through the blood-brain barrier after intravenous injection. One of the reasons that this dipeptide is interesting as a painkiller, an analgesic, is because its mechanism of action doesn't seem to involve direct binding to a G-protein coupled receptor. And yet, it definitely requires a membrane and signals across the membrane, and there is very good evidence that it activates inhibitory G protein, GI, the same one that is activated by the opioid receptors. Of course, many people are familiar with the opioid drug epidemic in the US, and there's a great need to develop drugs that don't exhibit the same problems with addiction. One of the issues with the opioids is that the opioid receptors have a number of desensitization mechanisms built in, and that desensitization is part of the mechanism of tolerance, which is part of the problem with current analgesics. So there is a computed folded structure for Kyotorphin inside a membrane. Actually, in aqueous solution, it's expected to have a fairly extended structure. This structure shows a direct interaction between the tyrosine and the guanidine group of the arginine side chain. This, of course, is a computed structure. No one has done a crystal structure of this inside a membrane protein. And this sort of highlights the need for developing small molecule models that can really inform both the computations and serve as models for spectroscopy for techniques, biophysical techniques other than actual crystal structure solution to tell what the structure of the arginine is. One of the major questions is what the protonation states are in this system. Here it's shown that both the guanidine being fully protonated and the tyrosine being fully protonated. But it's very difficult with any existing techniques to be absolutely definitive about that. And one of the things that I will show you is that some of the assumptions or conclusions that people have made in the past may be based on shaky experimental evidence. Okay, One of the, before I do this, I want to mention that already in the 1990s, we were starting to try to model guanidine side chains in nonpolar environments. We started with purchasing the ethyl guanidine molecule, which is just the tail end of the side chain of arginine. Ethyl guanidine carbonate and several other salts were commercially available for about $1 a milligram. And so we were able to do some experiments where we replaced the carbonate with other counterions, and we were able to measure various kinds of spectroscopic properties of these ethyl guanidine salts. But it became clear from the low solubility even of the ethyl guanidine salts in nonpolar media, the limit in chloroform was less than 1 millimolar, that we needed to find a way to get more of these compounds into very nonpolar solutions that could mimic the interior of membranes. The solution that became clear was we needed a longer side chain or something that was a bigger hydrocarbon group. And I was thinking, after doing an extensive search of what was commercially available, that I was going to have to embark on a synthesis program. But around the time I moved to Syracuse, several convergent factors made this unnecessary to start. One was better searches on the internet, okay, which allowed me to realize that even though long-chain alkyl guanidines were not available from traditional research chemistry suppliers, that they have a commercial use. And dodecyl guanidine, as it turns out, is made in quantities of millions of tons internationally. And I thought I would show this since this is an Agri-Food Biotechnology Institute. The area around Syracuse is used, has a big agricultural industry based on growing fruit, mainly apples and grapes. And so the use of dodecyl guanidine as a fruit fungicide made it possible for me to purchase several kilograms. This is a 2-kilogram bag of this fruit fungicide that is 60% dodecyl guanidine, and the rest is sort of an inert clay. And I was able to purchase this out of my own pocket at a local store for about $30. This gave enough quantities that I could just turn it over to both undergraduates and graduates in the lab and tell them to play with it and see if they could figure out how they could deprotonate it. It turns out that definitive deprotonation of guanidines in nonpolar solvents is a problem that it really turns out was never solved. Okay, Andrew Baniqua, a very persistent graduate student, finally figured out how to deprotonate dodecyl guanidine in 2012. He relied on standing on the shoulders of a group that had just recently, in 2007, published a crystal structure of just guanidine, CN3H5, free base. And they were so proud of their ability to publish this crystal structure that they basically said, they put right in their title, at last, the crystal structure of guanidine. And I was always shocked that the journal editors allowed them to actually put that in their title. But several of the keys to doing this were to make the bromide salt, the hydrobromide salt of guanidine, of dodecyl guanidine first, and to crystallize it in a very anhydrous form from acetonitrile. And then once these completely anhydrous crystals were formed, then to never expose them to water again, to dissolve them in alcohol and deprotonate with potassium T-butoxide, then evaporate off the alcohol, methanol, and to take the crystals, which at that point still contain a little bit of methanol, and again recrystallize them from acetonitrile. And this manages to remove all of the water. This was the first preparation that actually gives an elemental analysis that matches that predicted for the free base. So we haven't actually gotten crystal structures of those dodecyl guanidine crystals, but we've gotten some of some derivatives. And as I said, they all match the elemental analyses required to be certain that they have no water in them. So this work was published actually just this year by Andrew Benyikwe, who is now on the chemistry faculty at the University of Dodoma in Tanzania, which is his home country. So the first thing I'd like to show you is what the infrared spectrum of dodecyl guanidine freebase looks like. This is the structure of the dodecyl guanidine in aprotic solvents, benzene, carbon tetrachloride, dimethyl dichloromethane, and chloroform. The strongest band is a carbon-nitrogen stretch vibration, which is always between the range of about 1645 and 1651 wave number. And this is actually very different from what has ever been observed by taking arginine and trying to titrate it to high pH. One of the classic papers that looks at this is by a group, Veniaminoff and Kalnin, in 1990. They published a comparison of the spectrum in this region of arginine alanine at pH 5.6. This is the classic band shape of the protonated arginine side chain. By raising the pH to 11.2, they started to see this very, very clear band at around 1560 wave number, which many people, including myself, had thought for a number of years was characteristic of the initiation of deprotonation of the guanidine side chain. In fact, these spectra prove that that is not true. While we get the same spectrum if we take this high pH solution of guanidine, whether it's ethyl guanidine or dodecyl guanidine, and we extract it into chloroform, we get very much the same spectrum. The problem is that the extraction is pulling along at least one water molecule per guanidine, and that water molecule actually results in formation of the guanidinium hydroxide pair. So that this band here at 1560 is not characteristic of truly deprotonated guanidine. Instead, it's only characteristic of the guanidinium hydroxide ion pair present in nonpolar solution. It's also similar in aqueous solution. This corresponds then to a change between, in this case, a chloride salt of the guanidine, and in aqueous solution these would be not ion paired but actually completely separated. But at high pH, the solvated guanidinium, which is surrounded by water molecules, gets to be eventually replaced. At least a portion of that solvation shell must be replaced by hydroxide, which is capable of making stronger H bonds to the high NH groups, probably of two NH2s simultaneously. So there's two positions within this structure where it's likely that this hydroxide binds at high pH. And the pH that seems to be the midpoint for this titration, although it's traditionally in textbooks assigned at about 12.5, is actually probably closer to 13.7, corresponding to about 200 millimolar dissociation constant for hydroxide. And at that 200 millimolar, almost 1 out of 100 water molecules is deprotonated. So it's not too surprising that this stronger H bonding of the hydroxide is capable of displacing a water molecule from this first solvation cell and replacing it by hydroxide. And this is an even easier replacement in nonpolar solvents where there's no competition from, in aprotic solvents where there's no competition from the solvent itself. So this band is particularly characteristic, this band at 1560 is particularly characteristic of arginine hydroxide rather than deprotonated arginine. And this has prompted us to do some re-evaluation of some other experiments. Now, the results from the IR are also confirmed by NMR. So this is proton NMR, and we argue that this is the first observation of the chemical shifts of the protons on the guanidine group. So in benzene D6, we observe this at 3.56 parts per million. In DMSO, again, the free base, we see that resonance at 4.72. These peaks here correspond to an integrated area of 4 protons per dodecyl guanidine. And by comparison, this chemical shift value is much farther downfield in the hydrobromide salt, which can be made from this free base just by adding HBr, removing water, and then redissolving in the DMSO D6. An interesting aspect of these NMR is that you can't observe these chemical shifts for the guanidine group in the presence of even small amounts of water. They exchange very quickly, and all you see is a band corresponding to the water chemical shift. Even solvents that you think would work are problematic. So we don't show this in chloroform. For many years we thought and were confused, thinking that this proton resonance from the guanidine group was actually at around 6.1 ppm, which is actually identical with the chemical shift value for chloroform. And so even though we started with chloroform D, we would see a peak there that had an integrated area of 4 protons per dodecyl guanidine. No matter what concentration of guanidine we used, it would always rise to that level. And of course, if we use the pure chloroform D6, we saw just a tiny, tiny residual band at 6.1. Well, it turns out that that assignment was very confused because the protons from the dodecyl guanidine were actually ending up on the chloroform. And that's because this is such a strong base that it actually has an appreciable rate of deprotonating chloroform. So this we can show more easily on a time scale that is a little bit slower by adding chloroform to benzene. And in that case, this next slide shows some of the time-dependent behaviors. So we've just done some horizontal displacement of the spectra over successive times, shifting them to the right. And so you cannot see the protons of the guanidine group because they're so broad and down in the noise here. But they're actually, in terms of numbers of protons, what those 4 protons are doing is they are shifting onto the chloroform D and resulting in protonated chloroform rising with time. You can see this in this plot. This is the intensity of the protonated chloroform signal at 6.1 ppm, and it corresponds to just a little over 4 protons. Meanwhile, what starts out as 4 protons in pure benzene D6, once you add the chloroform, it drops very quickly within 5 minutes, even though this is only a few millimolar chloroform, and continues to drop. This is another thing that's quite surprising, was that when we actually analyzed this carefully like this, we could see that there's even catalyzed exchange of protons for deuterons on benzene D6, which is to say that there is an appreciable rate of base catalyzed deprotonation of benzene by dodecyl guanidine free base. This means that this is a much stronger base, I think, than almost anyone had ever realized. The pKa in textbooks of 12.5 is, as I said, corresponds only to binding of hydroxide. The actual deprotonation of this probably has a pKa of closer to 15 or possibly even higher. We can see this also from N15 chemical shift values, which have never been observed before. The dodecyl guanidine free base, you can see an iminoproton, a nitrogen corresponding to the imino group at 110 ppm downfield. This is the amino nitrogen at 49 ppm. This, of course, was measured with 90% N15 labeled dodecyl guanidine, which Andrew synthesized. And again, this is the HBr salt, which shows that you see only a single N15 peak corresponding to both nitrogens. These were synthesized with N15 labels only at the 2 terminal nitrogens using dodecylamine plus N15 labeled thiourea as the starting materials. Now, this separation of 60 ppm between the amino and imino protons is much greater than anyone has ever observed for arginine or other derivatives of it. And in fact, it again is much larger than the shifts that are occurring for arginine as you go through this titration at around pH 13.8. This chemical shift scale is referenced to a different standard than the one that we used in the preceding slide, so don't worry too much about the starting value being about 22 ppm larger. The main thing is that this only changes by about 22 ppm as you go through this titration, which is really only a very small fraction of what we see for a true deprotonation. So the conclusion is that, again, these bands, for example, at 1556 wave number, which we measured in my lab in 2004 using N15 labeled bacteria rhodopsin from Judy Hertzfeld's lab, we interpreted this band as being the largest band due to arginine in the time-resolved difference spectra of BR with its M intermediate state. So the positive band here, which is sensitive to N15 labeling, it changes between the green and the blue, means that this is a band that must be assigned to arginine. We always interpreted this as due to an arginine deprotonation, which meant that our estimation of what was the most likely proton release group in bacteria rhodopsin was somewhat in disagreement with what many people were feeling at that point. However, now that we have these spectra, we know that this is not due to a deprotonated arginine, but rather to formation of an arginine with a hydroxide bound to it. And this means that we're actually returning to a picture of the proton release group, which we first published. This is probably my most cited paper with Gobind in 1988, in which we drew the structure of M as having a hydroxide bound to arginine 82 and serving as the proton release group, this water molecule that was present in the L state, losing a proton to form this arginine hydroxide. This picture now actually corresponds more closely with what most people say based on X-ray crystal structures and cryo-electron microscopy. However, it's important to point out that although most people say that there are other groups involved in this, it seems that based on bands that you can see in the infrared, almost all of the proton occupancy change must reside within these two molecules, the arginine side chain plus the hydroxide. Well, anyway, we want to look a little bit now at the other protein, membrane protein that Gobind looked at was rhodopsin. And again, there is a highly conserved arginine, arginine 135, which changes its structure greatly upon formation of metarhodopsin 2. And without transducin, it was shown by this crystal structure from Klaus-Peter Hoffmann's group in 2011 that the arginine is quite close to a tyrosine. One of the nitrogens is within 2.7 angstrom of this tyrosine. What's most interesting that isn't often pointed out is if you look at the environment near here, it's almost hydrocarbon-like. Within 5 angstroms of this oxygen and this nitrogen, there is essentially nothing but hydrocarbon with the one exception of a methionine side chain. While this structure was able to discern numerous water molecules, several dozen in the crystal structure, none of them are within 5 angstroms of this tyrosine arginine H bond. In fact, the only other group that might serve within 5 angstroms as a possible H bond donor or acceptor from arginine is another tyrosine, which is slightly farther away. The interesting thing is that the distance between this tyrosine and arginine was perceived to increase in the crystal structure when the C-terminal tail of transducin was co-crystallized. And in this molecule, there's a carboxylate group which can serve as a counterion for that arginine and tyrosine. In this state, however, there is no obvious counterion other than perhaps the tyrosine. So the big question is, what is the structure of that arginine-tyrosine group? There's three general possibilities that you can imagine for sort of canonical representations of an arginine-tyrosine interaction inside a membrane protein. One is this first one, where both are protonated, and there is an external or a different counterion that is also present. And this is the one that looks like is probably present when you have the transducin C-terminus there, where the X minus here is the carboxyl group of the transducin. The other two are essentially the same charge, and they would be expected to be in rapid equilibrium with a proton moving back and forth between the arginine side chain, nitrogen, and the oxygen on the tyrosine. This is a sort of canonical, highly polarizable H bond, since these two are both have high proton affinities. These two structures are what we call the proton limiting structures for what we have nicknamed a Berry motif, and we use this abbreviation to stand for H bonded with a capital B, uncharged for the overall uncharged state, although in one portion of it it's a zwitterion, arginine and tyrosine. So the question is, can we model this at all? And in fact, computations that we did, these are density functional theory computations, indicated that if you actually put an uncharged guanidine near a phenol, that the hydrogen bond strength would actually be this huge value of minus 70 kilojoules per mole. Most of you who are biochemists may remember that an amide to carbonyl nitrogen hydrogen bond in a typical alpha helix usually has numbers associated with that H bond strength of about negative 10 kilojoules per mole. So this is actually gigantic, much larger than any of the H bonds that are present in base pairing in DNA. Now, for the dodecyl-linked version of the same grouping at right, the computation decreased to negative 20 kilojoules per mole. But we have synthesized, Andrew Benico synthesized versions of both of these, and so I can now show you some experimental results that test whether or not these are present. So the first thing is these are actual measured crystal structures of the dodecyl-linked phenol and guanidine. And this shows very clearly that you, instead of making that folded up structure, which we sort of hoped we would get, that internal H-bonded version, we form head-to-tail dimers. And the shortest H-bond distance here is between the nitrogen of one guanidine to on the neighboring molecule, the oxygen, and this distance is 2.7 angstroms, 2.75. This is pretty close to what was seen in the metarhodopsin 2 structure on the last slide. Now, when you have a shorter linker, this ethyl linker results in an S-cis bond, which is a slightly higher energy than the all S-trans form seen with the dodecyl linker. We think that this higher energy is tolerated and is made up for by having a stacking interaction between the nearby phenol groups, plus also putting the oxygen of this oxyanion at that sweet spot that I told you about, where it can hydrogen bond to two NH groups of the guanidine. This structure can be compared with the HBr salt, which, unlike the other two, can crystallize without any methanol. So far, we've only been able to co-crystallize with methanol, and what this means is that these structures actually don't show the maximum H bonding strength that is present in very nonpolar solvents. So in aprotic solvents, the best way to see this, that you actually form the same kinds of dimer structures, is by looking at the UV signal from the phenol group. So if you will turn your attention to these comparisons of spectra in DMSO and in methanol. So I'm going to show you a row of changes as you add guanidine groups to this paracresol molecule. So first of all, these are extinction coefficient spectra. So even though the concentration has gone up by a factor of 10 between the black and the red, the spectral band shape has remained the same. Now when we start adding dodecyl guanidine free base to this in these solvents, in methanol, you see the formation of deprotonated deprotonated cresol. And this is much the same spectrum that you would see if you were titrating this with sodium methoxide. This band at 296 is a well-known characteristic band of phenolate. In DMSO, there's a somewhat different absorption maximum. And I'm going to show you a little later on the concentration dependence of this equilibrium of this indicates that this is not a proton transfer in the traditional Brønsted-Lowry sense. This is a Brønsted-Lowry proton transfer in the protic solvent methanol. But here, what we're doing is we're making a Lewis acid-base complex. And you can see this most clearly by looking at the concentration dependence of the spectrum of this molecule. This is the one that I showed you a crystal structure of on the last slide. This is the free base of guanidine attached covalently to a phenol. And at very low concentration, this phenol is completely neutral. There's no sign of any ionization. And as you increase the concentration, you form this band at 318 nanometers. I haven't included spectra at higher concentrations than 10 millimolar, but if you go up to 200 millimolar, this 318 nanometer band eventually reaches almost equal in size to these bands to the band at 282. In contrast, in methanol, there's very little concentration dependence of the relative size of the protonated and deprotonated forms of the cresol. And this is classic Brønsted-Lowry behavior for a zwitterion. There's essentially buffering at the midpoint of the pKa's of the two groups. And we see similar things for the dodecyl link group. And the fact that the shape changes with the longer one, the deprotonated form is less favored for the long chain linker. That's a sign that the perturbation of the two pKa's on each other is smaller with a longer linker. Just as in amino acids, when you bring the amino group of an amino acid close to the carboxyl group, it tends to push their pKa's apart. The same phenomenon occurs in this case. You have pKa's pushed apart, and that results in favoring of the deprotonation of the phenol group here. Well, this effect is even stronger when you go to a less polar solvent. A completely nonpolar solvent of hexane is shown in here, and in this case you can see the band is at a little over, is also at around 315 nanometer, and these spectral changes occur over a range from just 0.06 millimolar up to 0.75 millimolar. These spectra are measured by taking solutions, 1 millimolar solution of cresol in hexane and about a 1 millimolar solution of dodecyl guanidine free base in hexane. When you mix them together at room temperature, they crash out of solution as crystals. And those are one-to-one crystals. You can take them, no matter what your starting stoichiometry is, those crystals have a one-to-one stoichiometry as measured by redissolving them in DMSO and looking at their proton NMR signal sizes. But if you take those crystals and redissolve them in hexane at higher temperature, around 45 degrees, you get these spectra that are shown here. Now let me go over this real quick. This black spectrum here is a measured spectrum of pure cresol in hexane. It's just sharpened. It's the same as we saw in DMSO and methanol, just sharpened up vibronically. All the other spectra shown, all the other colored spectra here represent one-to-one mixtures of cresol plus dodecyl guanidine that are made by redissolving those crystals in hexane. And if you look at the intensity of this band at around 315 nanometer and plot it as a function of concentration, this is on a log-log scale. Instead of increasing linearly with the concentration, it goes up as the second power. At low concentrations, the slope of this line is 2 on a log-log scale. And that's a sure sign that you're forming an equilibrium between two different molecules. You can also see this equilibrium shift by adding heat. So as you change the temperature, the lowest temperature is the highest spectrum here. As you raise the temperature, you tend to reduce the size of this peak. This is fully reversible. You can not really see it too well, but each one of these lines here of a different color is actually the superposition of two spectra measured first going up in temperature and then back down. And by fitting these using principal components analysis, we can figure out that the spectrum of the other component besides the black one is what's shown in red here. And this is not purely a deprotonated cresol spectrum. This is actually still a mixture of both proton transferred from the cresol to the guanidine and the other structure that I showed where the cresol retains the proton. So again, this structure represents the H-bonded structure that I showed previously. Yeah. Okay. So the H-bond enthalpy is so strongly dependent in the polar DMSO solvent. It's only minus 11 kilojoules per mole, but using the Van 't Hoff plot here, we get a value of minus 74 kilojoules per mole, which matches the computed value. So there's also results that show that the proton NMR structure and other slides that I won't have time to show you have this interesting property that this really strong H-bond results in very strange behavior for the proton NMR signals. So this shows the equilibrium that I showed you before, the formation of this dimer of the phenol with guanidine attached to it with an ethyl linker. And this equilibrium has a midpoint at a little over 100 millimolar. So measuring the NMR spectrum there, at low concentrations, the NMR signal from these guanidines is where I showed you before. It's very close to 4. As you go to the highest concentrations, it starts to approach the NMR signal from for the protonated dodecyl guanidine bromide. Meanwhile, although it reaches almost 2/3 of the way to that at about 100 millimolar, the change in the paracresol signal goes only a fraction of the way towards its values in the potassium cresolate salt. What this means is that you can't rely on NMR to tell you reliably when these groups are protonated, not nearly as reliably as you can by using optical spectroscopy such as UV or IR. When the H-bonded dimer forms in an aprotic solvent environment, both the guanidine and the phenol look mostly protonated by NMR, even though they are sharing one proton between them, and overall the pair has been deprotonated. So getting back to here, in this structure, we speculate that this very strong H-bond between tyrosine and arginine plays an important role in the activation of G-protein coupled receptors. There's an energetic problem which people may not realize of having a buried arginine in such a nonpolar region of the protein. The problem isn't necessarily so apparent when you think about this reaction starting from rhodopsin, because you're adding the energy of a photon, and you might imagine, well, you could end up with quite an energetic state at this point. But you also have to remember that this reaction goes also in the reverse direction here. Metarhodopsin 2 with a G-protein tail has that tail being removed. The counterion of this C terminus is being removed, leaving a naked positive charge, if that's what it is, in that very nonpolar binding site. And the best explanation for how that can happen is if this C-terminal tail pulls a proton with it, leaving this H-bonded uncharged arginine tyrosine that has a very strong negative 70 kilojoule per mole H-bond between it. And so these are just quickly the conclusions. The arginine side chain binds hydroxide very tightly. The only accurate way to model the fully deprotonated arginine side chain is in aprotic solvent, free of water and hydroxide. All prior measurements of arginine titrations in water showing a pKa of 14 have actually measured a hydroxide binding constant corresponding to a dissociation constant of hydroxide of about 200 millimolar. And in hydrocarbon-like environments, a phenolic group can form an extremely strong H bond to deprotonated guanidine with a formation enthalpy as large as minus 70 kilojoules per mole. This could hypothetically occur in membrane proteins as this Berry group that we define. And detecting this group in proteins should probably not rely on NMR, which can give this deceptive and anomalous result where both groups simultaneously seem protonated even though one proton has been removed from them. The rapidly exchanging shared proton might appear to have an occupancy of greater than 80% on both guanidine and phenol simultaneously. Thank you for your attention. Oh, and I just want to put some acknowledgments up there. Thank you.
Thanks so much, Mark. Is this on? Yeah. Thanks very much. That was a very, very detailed biophysical talk. Very nice. It's good that we have a broad range of subject here. We are running a little bit late. Is there any, maybe one question? Has anyone got? Can I just ask a question that occurred to me? Because how general is it? I mean, you must have have you looked in crystal structures where this sort of constellation of tyrosine, arginine occurs? I mean, is it very unusual? Or
so there's an atlas. There's an atlas of crystal structures that was first published in the early 1990s, and at that time there were a few that seemed to have this constellation of possibly a linear arrangement of arginine with tyrosine. And they've gotten a little bit more common in that sampling of crystal structures, which is now online and is updated regularly. Okay. So there does seem to be, so they cataloged all the pairwise interactions between different pairs of amino acids. And for arginine and tyrosine, there's about 5 or 10% of the arginine tyrosine close interactions within say 5 angstroms of each other. About 5 to 10% of them seem to have this kind of...
And does it have a special role in biology? I mean, do they always have this sort of... Because it's such high energy, that they have a specific role in each case?
Well, I did not go through the entire database and look at all of the examples. The crystal structures of the GPCRs that have been solved do show conserved, but not necessarily with this close a distance for this H bond of 2.7 angstrom. This is shown most clearly with the metarhodopsin 2. Again, we are speculating, we're hypothesizing that this does play, in the case of GPCRs, a key role in terms of being able to form this very stable group which can attract into the interior of the membrane the C-terminal tail, which is an acid group. So buried inside the membrane, this arginine tyrosine, if it's neutral, still has the quality of being very basic. And so this would explain how the C-terminal tail of it would help to explain why the C-terminal tail of transducin is so attracted to that point, but also how it can then be released without violating free energy principles.
That's really interesting. Thank you very much. I think we need to move on now. Please join me to thank Mark again for his wonderful insight.
Thank you.
So we come to our last speaker. Let me just quickly get my notes. This is Professor Yotaro Takagaki, is that right? He got his PhD in biological chemistry in 1975 at the Faculty of Agriculture, University of Tokyo. He was a postdoc at the National Institute of Genetics in Japan with Hirota. Then he did his second postdoc in Gobin's lab between 1976 and 1982. Moved then to the MIT Center for Cancer Research, where he was staff scientist, went back to Japan in 1990, where he worked first at the Mitsubishi Kasai Institute of Life Sciences in molecular immunology, and then at Kizar Kitazako University Medical School, where he was a professor, and then between 2005 and 2010 he was at the Tokyo Women's Medical University and is now visiting professor at Nikon Pharmaceutical University since 1913. And the title of his talk, Gobind in the History of Protein Sequencing. Please, Professor.
Thank you very much for kind. Thank you very much for kind introduction. I'm sorry, lots of difficult Japanese names. It's already 40 years, more than 40 years since I joined Gobin's laboratory, so it's a very old story. I'm basically retired after this job and writing reviews and that sort of things. I volunteered to talk in tomorrow's session, but found myself in the main lecture, and I was surprised, but I went back to my old notebooks and tried to find things to talk, and so it was, I hope I could give students here some of the message that I want to convey. Okay. Gobin, in his difficult time, he said in a movie previously, around 1951, he actually produced a paper for stepwise peptide sequencing from N-terminal. It's a thioacylation method. Edman came out with isothiocyanates in 1950s, and 1 year after Gobin's thioacylation method came out, this had an advantage of producing free amino acids. And so some people did. Okay. Thank you very much. Okay, thank you very much. So, some people took, like, there's a book concerning advanced method in protein sequencing. Russell Doolittle of UC San Diego used this thioacylation method until this time. In this book, he mentions Kenner and Corona paper. This is 1952. And then he describes in this chapter how he modified thioacylation method. But anyway, his Gobin's sequencing method persisted quite until recently. Although Edman had an advantage these days because of high sensitivity of HPLC. HPLC made possible the Edman degradation below sub-picomolar level sequencing. So Edman degradation has been a major method, but Gobin's method still persisted and quite a long time. He also published paper concerning stepwise peptide sequencing from C-terminus using carbodiamides. And there were other chemical reactions too, but the chemical reaction has a drawback. If you meet with proline, then sometimes sequencing gets blocked, etc. And then there are much easier method of carboxypeptidase method, sequencing from C-terminus now. So this didn't take on, but Gobin, in his beginning of independent career, had desire to get into proteins. So when I joined in 1976, actually, this is Yuki Hirota, my boss for my first postdoc. He discovered DNA polymerase 2 and 3s. he told me that you should associate yourself with great person for your scientific development. And he was very helpful in squeezing me into Govind's laboratory. This is Marcia Rosner when she was young. And then this is Mike Ryan. Marcia is still young. Sorry. The last person to work with tRNA synthesis function, he was pursuing function. But when I joined, Govind told me that in 1968, in International Biochemical Society in Japan, I said to people, I'm going to synthesize a functional protein in vitro, in glass tubes. And most people, almost all people said, you are crazy. You are stepping into the region of God's area. And he smiled, but I've done it. And then he told me, proteins are important. Membrane proteins are very difficult. So I'm going to work with membrane proteins. That's his attitude. It took me 6 years to complete what I had to do. At that time, he didn't take on thioacylation sequencing method. He actually used Edman degradation because Gerhard Garber, he was a great guy. He's a nice person. He came from Canada, McMaster University. He sequenced elastin before coming to Govind's laboratory. So he's basically a protein sequencing person. And sorry about the quality of picture, but this is the Beckman 890C spinning cup liquid phase protein sequencer that Govind's laboratory had. Spinning cup, well, actually, if you look at this portion, there's a cup rotating, and aqueous liquid, sorry, I'm not used to this one. Aqueous liquid film is formed here due to centrifugal forces, and then organic solvent gets delivered here, and organic solvents skim surface of an aqueous phase, delivering reagents and then collecting products, and then gets siphoned off to test tubes. This is a liquid phase sequencer, automatic sequencer designed for Edman degradation. And Govind's group, this is the first amino acid sequencing project of Govind's group in MIT. These arrows indicates where Edman degradation can decipher, but there are areas that this Edman degradation cannot cover. And in chemistry department, Klaus Biemann came to us, and he said he's going to sequence any protein with mass spec, mass spectrometry. And Govind, I think he told me later, but Biemann says that he only needs a few milligrams of proteins. I think we ended up supplying Klaus Biemann's group a few 100 kilograms of bacteriorhodopsin. It was culturing bacteriorhodopsin. Chris Clay cultured so much that our servo centrifuge was clogged up with sodium chloride crystals. We take out the mortar, put it in water, and then dissolve the salt, and then put it back again, and that sort of things. But anyway, mass spec was used to cover the area that Edman degradation cannot cover. Wait a minute. However, Klaus Biemann's group started sequencing proteins with mass spec. At the beginning, it was gas chromatography mass spectrometry, GCMS. The first one was 1976, 44 amino acid, monelin. And then second happens to be bacteriorhodopsin, 248 amino acids. And if you look at Klaus Biemann's reviews, there are several reviews. He always mentions about and thanks Govin Khorana for providing bacteriorhodopsin. But then he went on to sequence E. coli alanine tRNA synthesis. I think Datazo's name was mentioned in this description. He was somehow instrumental in providing, I think, protein samples. But then in 1981, fast atom bombardment MS came out, so GCMS was no longer available. These days, we use MALDI or matrix-assisted laser desorption ionization method is used for protein sequencing. But actually, it was in competition with a Russian group. And so there were areas that mass spec couldn't cover. And in fact, we got help from a solid phase sequencing company called Sequent. And because I used to live in Brookline, and this company was in Watertown. So when I commute, I kind of became messenger boy and delivered the peptide and delivered back the result and that sort of things several times. Alex is being a very kind person, and I think he did sequencing. He helped us sequencing free, I think. But then Govin eventually bought solid phase sequencer from Alex. Also, Edman liquid phase sequencer was used to locate the retinol position. I was the operator of a sequencing machine after Gerhard Garber left. One important thing that I want to mention is that we kind of developed organic solvent system. This is gel permeation, gel filtration chromatography, ethanol, formic acid, 4 volume of ethanol and formic acid 1. We leave it for 1 week, and eventually ethyl formate is formed and equilibrated. So it must be a mixture of these four components. This was very great solvent for solubilizing membrane proteins, very hydrophobic proteins. Now getting into main topics, I'm going to talk about the photocross-linking sequencing that I did in Govin's group, because this hasn't been talked much. Govin's group developed many phospholipids with photoactivatable probes, but we ended up using carbene generator. Carbene is carbon losing 2 covalent bonds. And this has a capacity to associate with organic compounds like hexane, CH insertion, for instance, upon photoactivation. Another one was diazo trifluoropropionyl group. Dr. Chitar Gupta over there synthesized this compound, labeled this compound with C14. And I didn't know until a few hours ago that when he synthesized these radioactive compounds, the firefighters and radiation protection officers were standing by just in case explosion happens. Because he used radioactive phosgene. So I didn't know that until today. Diazo, okay. So it was a very dangerous project, to say least. But unfortunately, this diazo group got reduced by dithiothiol or beta-mercaptoethanol reductants. And so I looked for the protein that doesn't have any cysteine for sequencing project, and I found cytochrome B5 in Dayhoff's protein atlas. It has a C-terminal hydrophobic domain, which gets, usually it's octamer aggregate, but if you mix this with phospholipid vesicles, it spontaneously inserts into these vesicles. If you have other type of membrane like mitochondria, then this moves to another vesicles. So it has a capacity to transfer between vesicles. And if you apply carboxypeptidase, then it eventually loses the ability to bind to phospholipid vesicles. These are called loose binding form. However, if you treat this preparation with dilute cholic acid or sonication, then somehow the samples become no longer transferable, and carboxypeptidase doesn't digest, it stays on the membrane. So it's called tight binding form. And this square is supposed to be hydrophobic sequencing. Govin's second sequence project was sequencing of this hydrophobic region of cytochrome B5, which was published in 1980. So we tried to see the membrane embedded portion using phospholipid-containing photoactivatable probes. And to do that, we checked into the conditions. Finally, we ended up using an arc lamp, 1,000 watts, mercury xenon arc lamp, but we passed the light through the monochromator, and then to make sure that we don't get any stray lights into it, we purified the light with filter cutting of 345 and 360. The sample, actually, there is a cuvette here, the light passes through, but the sample is covered with water jacket to cool it down, and also the sample was under strict nitrogen. If you shine the protein with oxygen in it, without any photoprobes, these lights will somehow make aggregates of your proteins. The oxygen causes activation of maybe tryptophan, something like that. And it's known that you have to shine the light to protein without oxygen. Gupta's reagent, it took 40 minutes. Radha's reagent, the top one, took 2 minutes. Nevertheless, Radhakrishnan and Dr. Gupta both made the vesicles and photo-irradiated their samples and measured the point of distribution of crosslinks to opposite strand using mass spec, and the beautiful pattern of bell shape was, at least this is a part not complete, but was obtained. If you label here with a deuterium and measure the distribution of these deuteriums, it scatters around like this. So it's reflecting a similar type of distribution. it looks like having a benzene here, phenol group here doesn't interfere the movement. So I did the cross-linking using these conditions just to make sure I added the tryptophan 25 millimolar into the solution too, to avoid tryptophan damage. This is a gel permeation chromatography, phospholipid monomer, phospholipid dimer, cross-linked cytochrome B5, but cytochrome B5 also got oligomerized, likely to be oligomerized at the hydrophilic portion having heme inside. And this cross-linked portion was treated with alkaline and acid. There were a huge amount of, or more than half of acid label, alkali label, cross-linked product. And these label products cannot be analyzed by Edman degradation or stepwise sequencing, because stepwise sequencing requires acid phase and alkali phase. So by stripping off unstable groups and then applying them to the sequencer, but liquid phase sequencer couldn't retain the cross-linked peptides. It just went to organic solvent. So I had to use solid phase sequencer. This is a solid phase sequencer that Govin bought. Also later on, Alex Bonner, I was kind of involved, but changed this to DNA synthesizer too, using Marv Crothers reagents. And also, I had to improve the coupling yield. Aminopropyl grass speeds, control poor grass speeds, didn't couple very well, only 5% or so. So we, with Gupta's suggestion, we lowered the pK of amino groups by having a allyl group here. And this new grass speeds coupled more than 50%. Using this, also the sequencer has to be tuned up to get higher repetitive yield. Edman degradation, each step is not 100%. It could go up to 97%, but not more. And it's quite difficult to get up to 97%. I changed the tubings and lots of things, programs, etc., to get to 95% yield. So this was original cross-linked product coming out of machine. I corrected the value for repetitive yield in dotted line, and these are the distribution of cross-linked area, starting from serine here, serine here, serine here, serine here. Many times it was the same. So we sort of with that result, we kind of speculated the membrane-bended area of loose binding form, and this was figure 7 of my JBC paper, first paper. This was taken up by textbook. It says permission of author, but they never asked our permission. So tight binding form. I did the sequencing same, but the sequencing area, cross-linked area was somewhat different. It started from this serine here, 3 residues into the hydrophobic area. These are very hydrophobic peptides. And so difference is loose binding form is wider, tight binding form is a little bit more compact. And to cut the story short, to answer the question, what is a form of C-terminal, and I created unsymmetrically distributed vesicles having C14 in outer leaflet only using phospholipid exchange protein of Carl Oates. And then also I made a tritium-labeled phospholipid predominant in inner leaflet, taking out the outer leaflet radioactive protein, diluting out the outer leaflet phospholipid, photo-labeled phospholipid by adding acceptor here, and cross-linked separately and mix them together and use the chemicals to get the fragment, and CO ratio clearly showed higher CO ratio at the N-terminal side with a gradient of going down, which is consistent with transmembrane configuration. Now, if you look at the protein sequence of this hydrophobic region, there's a proline here. And proline has a very low threshold of cis and transform threshold. So my speculation was that loose binding form has two helices in outer leaflet with proline in cis form, but then if you sonicate or if you treat it with dilute detergent, then this becomes transform, and the helix comes much more tightly, and so extra cross-links that was required is no longer cross-linked, and this is the farthest that I could get. Gobi asked me to make CPK model whether the speculation fits, and it does fit quite nicely. I'm out of time, I think. So recently I'm doing proteomics using MALDI-TOF and that sort of things, but I'm not going to get into this, except there are, well, I did put myself in genome research, molecular biology at one time, but membrane protein analysis is very difficult, tedious. Easy things get done very quickly. So for young people, don't put yourself in easy things because you lose a job in 10 years' time or something like that. Things will change very quickly. Always try to work with important projects, but difficult ones. This is an example of, if I do analyze the proteomics of many interesting samples, like this one, cardiosphere, it's cardiac stem cells. We see huge amount of heat shock proteins. They are more than half of the expressed proteins. So, and if I analyze, this is a heat shock protein with this different charges. The antibody against phosphotyrosine, antibody against acetylated lysine, those will show you that these are chemically modified materials. There are huge area that needs to be analyzed of post-translational modification. And that's something that we have to, heat shock protein, it must be responding to the environmental change or some condition. These are the areas that will be, I hope will be flourishing in 20 years' time, something like that. I associate myself with Nobel laureate of physiology and medicine. Osumi actually happens to be, he got his Nobel Prize with autophagy last year. He was 3 years senior than me. For my undergraduate thesis, he actually was a tutor. And then after Gobin, I moved to Center for Cancer Research as a staff scientist. Salvador Luria director then provided me machinery that I needed. And then basically I associated with Susumu Tonegawa. We discovered T-cell receptor alpha chain, gamma chain, and gamma delta T-cells. And then Phil Sharp succeeded Salvador Luria in 1985. He immediately came to me and asked me to open up a microchemical laboratory in cancer center. I used to produce 80 oligos per month provided. And I'm sure that that was quite helpful to become MIT biology department as #1 ranking in United States at that time. The important thing is that these people has very common character. They stick to something important. They are single-minded persons, but stubborn persons. They are persistent and never jumps around. So look for a great person who has insight, very consistent with the patients. Unfortunately, Tonega wasn't a patient person, but other people were very patient. But my Govin used to say dead center focus, and that's what these people did. One thing, big advice, I associated with Govin 6 years and Tonega with 7 years. That's a golden step for failure. I mean, one should get independent much earlier to grow. And association with great people for a long time is not that good. Thank you. Thank you very much for letting me talk all the stories.
Thank you very much for your very insightful pieces of advice. Does anyone have any comments? I know we're getting very late, and this is a very historic talk, but does anyone have any comments? But if not, please join me to thank you once more for your wonderful talk. Okay, so I head back to the organizers now.
Yeah. Thank you very much, everyone. We would now request all the delegates and the poster participants to please go to their posters as there will be evaluations, and then we will resume for dinner at 7:45. So we close the session now. Thank you very much.